Tuberculosis is a chronic bacterial disease which was considered as a very important disease in domestic animals and in man that before discovery of tuberculin and BCG. This disease is a granulomatous chronic disease characterized by the development of epithelioid cell granulomas or tubercles. The cause of this disease is three species related to genera Mycobacterium. First is Mycobacterium tuberculosis, which affect man but can affect animals, particularly cat and dog. Second is Mycobacterium bovis, affecting domestic animals but can affect man, particularly children, giving rise to special forms of tuberculosis, lymph node, intestinal and bone tuberculosis, and Mycobacterium avium, which affect birds but can affect occasionally man and very rare domestic animals, but can produce local lesion in swine and horse. The pathogenesis of tuberculosis classically is divided into two periods: period of primary infection and period of post-primary infection, related or according to a different pattern of reaction of the host due to second exposure to the bacteria. We begin with the study of discussion of period of primary infection. The period of primary infection include the development of primary lesion in the organ and the development of primary complex, including the regional lymph node. We speak first about the development of primary tubercle, or primary granuloma of tuberculosis in the organ. The tubercle at the site of entry is taken by a macrophage and bring the bacillus or the bacteria in the tissues. As soon as in this macrophage the organism can live and replicate in the macrophage. When reach the tissue, the first cell reacting is the neutrophil. It counts a group of neutrophil which cannot eradicate or kill the organism. They die after degranulation and disappear. The multiplication of the organism in the tissue attracts more macrophage. The macrophage engulf the organism, digest the organism, and present the antigen of the organism conjugated with H. Conjugated with MHC class 2, which is the major compatibility complex class 2 found in antigen-presenting cells. Presented this to the CD4 lymphocyte, which release chemotatic activity, activating and lymphokines such as interferon gamma, which activate and attract more phagocytes or macrophages. These macrophages can differentiate or mature to epithelioid cells the epithelioid cells are less phagocytic cell, that the capacity of phagocytosis of epithelioid cells is less than the macrophage, but they have more capacity for intracellular bacterial killing. Also, some of the macrophages will coalesce together and bind together and produce Langham giant cell. By this step is formed the picture of the young tubercle, which is usually surrounded with a zone of lymphocytes and undergo central caseous necrosis. The necrosis of the center of the young tubercle is due to many factors. First, the lysosomal enzymes and the tumor necrosing factor and the oxygen active free radicals, which are released by damaged activated macrophages, and also due to increase on accumulation of the mycobacterium byproducts, and also due to the hypoxic condition of the center of the tubercle. After the development or during the development of the primary tubercle, the organism can escape in a macrophage or free through the lymphatics and reach the regional lymph node, producing a tubercle or granuloma in the regional lymph nodes. In this case, the primary complex is well developed. The primary complex is found in the lung in 90% of cases of tuberculosis in cattle, in 40% in calves, in 50% in dog and cat, and it is rare in swine and not found in birds, but otherwise it is in the digestive or the intestinal tract, the digestive system. The presence of what is the fate or the outcome of a primary complex? We have a primary complex forming. Either produce healing, يحصل healing. Healing يعني إيه? Means that the organism is eliminated, killed and eliminated, and the tubercle in the regional lymph node and in the organ is transformed to a mass of connective tissue, but leaving hypersensitivity of the organ or the tissue to the organism. And there is a hypersensitivity. A second probability is that dormant tubercle. The tubercle is encapsulated in a connective tissue capsule, and the organism is found in the capsule inactive. This is a dormant tubercle. Flare up under unfavorable condition and when the resistance is decreased. Another possibility of outcome of the primary complex is tuberculous pneumonia or tuberculous bronchopneumonia. In this case, the organism expand in the lung and produce secondary tubercles. And sometimes a tubercle, when break a bronchus, the infection reach the bronchoalveolar area and produce tuberculous bronchopneumonia. This means that the third probability is the development of tuberculous pneumonia and tuberculous bronchopneumonia. When the tubercle break in blood vessel, or the organism is reached the blood through the lymphatics, the blood is seated with the organism, which is distributed all over the body, trapped by capillaries in different parts of the body, and produce early generalization with the development of miliary tuberculosis, characterized by evenly distributed tubercles in the same age and same side, showing advanced caseation and calcification. This is the period of primary infection. We speak about the period of post-primary infection. This period is observed in animals which survive the period of primary infection. In this case, there is two probabilities: either superinfection or second infection, and this is endogenous or exogenous, and it is usually in case of lung exogenous, or break up or flare up of a dormant tubercle producing secondary generalization. The superinfection or second infection produce a peculiar form of tuberculosis called the chronic organ tuberculosis or isolated organ tuberculosis. This form of tuberculosis is characterized by stopping spread through blood or lymph, stopping infection of the regional lymph node, and the regional lymph node not sharing in this second period. Also, no calcification, but excessive necrosis or caseous necrosis or softening and excessive proliferation of granulomatous tissue. This form is more observed in the lung or the mammary gland of the cattle, and is also observed in man. There are three forms of chronic organ tuberculosis in lung, which are: nodular form is consisted of different nodules, different in size, consisted of a central necrosis or caseation and a peripheral big zone or excessive granulomatous tissue. The second form is caseous form, which is characterized by caseous necrosis, which according to spread can be acinus, lobular, or lopal. The third form is the cavitation form. ولا ده مش مهم؟ خلاص يعني ممكن يسجل. is due to liquefaction of nodular or caseous necrosis, and this is called liquefactive cavitation. Or there is disorganization of the wall of the bronchus leading to dilatation of the bronchi, and this form is called bronchiectatic cavitation. In the period of post-primary infection, when under unfavorable condition and the resistance of the animal drops, the organism become active, pass through the capsule and spread, producing secondary nodules. When the organism reach the blood through a break in a blood vessel or through lymphatics, it produces late generalization. The extrapulmonary tuberculous lesions in different organs are, for example, in liver, nodular, miliary. In kidney, nodular, miliary. In uterus, nodular. In intestine, ulcerative and nodular. In the serous membranes, pearl disease is the presence of evenly distributed tubercles resembling the pearl in distribution and in general features. That is pearl disease in serous membranes. The species variation, while the principal features of tubercular reaction or tubercular lesions is the same, but there is species variation. For example, in cattle, we find that the lesion, the primary complex, 90% are found in the lung. It means that the respiratory system is the main system affected. Also, there is caseation and calcification. While in the horse, the main lesions are found in digestive system and spleen, and there is no tendency for caseation or calcification, but only extensive proliferation of granulomatous tissue. Also in dog, in pulmonary tuberculosis, we find diffuse epithelioid cell diffusion of epithelioid cell distributed in the lung with discrete tubercles and absence of Langham giant cells and no tendency for caseation and calcification as in horse. خلاص. Paratuberculosis and Jones disease is a chronic bacterial disease characterized by chronic enteritis, diarrhea without blood or fetid smell, also with the thickening of the intestinal mucosa and the corrugation of the surface of the mucous membrane. The cause is acid-fast bacterium known as Mycobacterium paratuberculosis with 1 bovine strain and 2 ovine strains. Sheep and goat, and the mode of infection is principally by ingestion of contaminated food or water. The disease has a very long incubation period and a long course. The clinical manifestation do not appear except after 2 years old. Clinically, the animal become emaciated with obstinate diarrhea without blood or offensive odor, and also sometimes in some cases there is edema of the submaxillary space which disappears after the onset of the clinical manifestation. The organism can be observed by or detected by immune fluorescent technique in the feces before the appearance of clinical manifestations. After infection by ingestion, the organism replicates in the tonsils, the suprapharyngeal lymph nodes, the mucous membrane of the intestine, and the mesenteric lymph nodes. can pass to the liver, producing its lesions, and in pregnant animals, the organism can reach the embryo or the fetus by intraplacental passage. General infection with the organism is rarely or occasionally observed spontaneously and in experimental infection. The change in the intestinal mucosa is mainly in the last part of the ileum, but it can be found at any part of the intestine, small or large intestine. The changes begin with the accumulation of lymphocytes, plasma cells, and many eosinophils, and few epithelioid cells. Later, the epithelioid cells increase in number with the proportionate decrease of the number of other cells. The epithelioid cells in this disease are big cells with plenty of cytoplasm, with sometimes multinuclear cells are present, and when stained with Zee-Nelson, it will be found that the epithelioid cells are crowded with acid-fast bacilli. In advanced cases, the mucosa propria is packed with such cells which press on the crypts of the gland or causing their obliteration, leading to cystic dilatation with progressive glandular atrophy. Also, the migration or the reach of these such cells in the submucosa can happen, but leaving the muscularis intact and free of the cells. The lymphatics in the intestine are surrounded by such cells, lymphocytes, plasma cells, and epithelioid cells, with the development of an epithelioid cell granuloma in the wall of the lymphatic, which projects in the lumen of the lymphatic, causing its proliferation. And this is mainly the cause of the swelling and the edema of the mucous membrane of the intestine in this disease. In cattle, there is no nodules, no calcification or caseation, but such changes can be observed in sheep and goat.